Okay, so yeah, so I've been talking about geometrical methods for biological networks, but first of all, I should, of course, thank the organizer for having me here. I hope I won't be, I give this charge as I'm a mathematician, so I hope it won't be too mathematical. And I would like to thank Indika for hosting me and for proposing so many interesting problems and so many figures he has in mind. The better figures in the slides are his. Okay, so what I'm talking about, well, I won't go into depth. Just a very bird's eye view of things. So it means that I won't go into any technical details. Huh? Okay. So what next? No? Okay, now it's okay. So I won't go into details. That means that at some point I might be rushing or skipping all the stuff. I try to keep things, in time and cover as much as possible of things that might interest you. And on the other hand, so I try to be user-friendly, no, not too much mathematics. At least that's how I think I try to do it. It's not sure I succeeded. On the other hand, I was concentrating, as one example or so. Everything else is from biology, all the examples. Biology of sorts. And on the other hand, I won't pretend I'm something which I'm not, so I wouldn't dare to bring also Athens and try to explain too much biology to this audience. So excuse me if I make mistakes here now. Okay? Saying goes, so the owl was Athens' holy bird. So having birds to Athens means doing something futile. Okay, so a very brief introduction. Why networks? Well, we know we have social networks, we have biological networks, like gene interactions, neural activities in cortex. We have transportation networks. We do networks in architecture for 70 years or so. So they are also important for computer science. So this is just an overview. So till recently, people were talking about networks and graphs are the same, but so all the measures were coming from either from computer science and node measures like clustering coefficients, so on and so forth. So more recently, people started to understand that network doesn't mean just the nodes, it means the relations, first of all. So relations mean edges, and edges you need another type of curvature or other type of measures, totally a different type of measures than the measures people use for nodes. So one measure we'll use is a Ricci curvature, and I'll talk more about that. So when you talk about higher order correlations, not just edges, you can look at triplets and quadruplet points correlating and so on and so forth. You'll see more examples. Then you talk not just about graphs, but more complicated objects that are called polyhedral or simplicial complexes. So it's a bit of mathematics so far, but I will forget it soon. We will get more light. So in the beginning, people were doing combinatorics. Everybody knows how to count. And at some point, they started doing this function analysis, metric spaces trick, embedding in L infinity, which is cool. We have a lot of nice mathematics, but come on, it's not, it's infinitely dimensional. And so there are a lot of Laplacian-based methods, but this also has some limitations. So people started looking for geometric methods, more expressive. So what geometric tools? Well, curvature, first and foremost. Why? Because it's not, perhaps the name is new to you. I try to suggest that it's not, in fact, but it's very intuitive. So for surfaces, it just means, this is a good example. So this little prince is on a small planet. It's very curved. The horizon is very close. I live on a big planet. For me, the horizon is far, far away. I think everything is almost flat. But it's very intuitive. So on the more higher level take, why should we do curvature? Because the main notion of differential geometry is that of curvature, which is the same of regretted Robert Brooks. And I would go even further and say that basically all geometries basically looking at curvature in a sense or another. Okay, So let's go back and see what curvature might mean. So it's an idea which goes it's very intuitive. A straight line has zero curvature, of course, it's not curved at all. So it's zero curvature, straight line. What's the next more complicated figure which you learn? Well, the triangle, but triangles are kind of not smooth. So we first learn about circles. So the Greeks already knew what the curvature of a circle is. It's 1 over the radius. The smaller the radius, the more curved the circle. So this is it. Oh. It's very intuitive. Again, another picture of the same. And then you think, okay, so this is a circle. Can I do for surfaces? So for surfaces, the idea which goes back before Gauss, more or less, is like this. You have a surface and you cut it with a knife. Introduce a nice perpendicular in all the possible directions, okay? So let's think this is a piece of bread. If I cut it like this, okay, don't try it at home. But if I cut it like this, I get a small piece of bread. It's good for tartines with caviar or something. If I cut it this way, I get a Subway sandwich. In between, I get all the pieces of bread possible, yes? So in each direction, I have a curve. Each curve has a curvature. It's intuitive. Somehow you think of it as curvature of the circle that approximates is the best. Okay, as long as it's not mine. So you have infinitely many curves, infinitely curvatures, but there is a beautiful result, which you, of course, I'm sure Indigo appreciates. Linear algebra tells you there is a direction with maximal curvature, one direction minimal curvature, they're always perpendicular. So if you multiply these numbers, these curvatures, you get the Gauss curvature, which is the curvature for curves. Okay, so now what I want to say before we pass to biology is you might think you never heard of this, but in fact you know this from the 8th grade, at least. When we learned in the 8th grade. You talk about parabolas x squared. So the more, so it's, if you see, I'm looking at function x squared, the derivative at 0 is 2. The function is 2x squared, the derivative at 0 is 4. And of course, the curve is more steep. If you take half of x squared, the curve is more flat, and the derivative at 0 is 1. And so it's, in fact, my mathematician friends would probably kill me, but you can think of curvature just an upgraded posh version of the second derivative for more complicated objects. And yes, we have the intuition. Never mind the 8th grade. You see, we use this notion of curvature every day. There is a champagne glass, it's more curved, more pickier than the red wine glass or a brandy glass or that. Okay, so now, since we're in a good mood, there is a problem with this approach. The problem is that all this stuff which I draw, or try to draw, pretending to draw, it's smooth and nice. And it's far away from being a graph. Yes, this is just smooth objects which are usually graphs of functions like this thing which MATLAB produced. That's it. Sorry. I'm forgetting you don't see my laptop, but my screen. So the idea is not to do that, but to do something else, which means to discretize. Right? So one instinct that people who work in computer science, lithographics, imaging, they approximate. But there's nothing to approximate. When you have a graph, a protein-to-protein interaction network, it doesn't approximate anything smooth. There is nothing to be approximate by it. So when you want to capture a notion of curvature, you have not to approximate, not to lie, of course, but rather to discretize. That means take a property essential to the notion that you want to capture and try to implement in a discrete setting, in a graph setting, or polyhedral setting, whatever. So this is discretization, and there is more than one way of doing it. So there is fun in looking for this discrete analogs. On the other hand, it's also a cheap way out, because otherwise you would need something which, please don't faint, it's called tensors, and we don't like that. Okay, so what is Ricci curvature? So I talked about ordinary curvature, so the notion of curvature which best fits networks is that of Ricci curvature, because associated to directions, and the graph direction are edges. We said we want to look at measures of edges. Okay, so it measures two things: how far volumes grow. So here are the volumes. Increase the radius, but it may be of a cone, but it might be the radius of a ball. And how far geodesic, i.e. straight lines, or shortest lines, straight lines, diverge. So things of a group of soldiers, they start together in a direction, but then they go a wall. Each may reach together a target, or they get lost, or go lost, and then meet again at a point. Yes, we meet here and there in 5 hours. Okay, so how can we understand that? When so for positive curvature, the phenomenon is captured by the sphere. So if you start from the North Pole, the straight lines are the geodesics, the meridians. So they start, they go apart, and then meet again here. So it's an Auf-Wiedersehen kind of theorem. You have to meet again. So that's positive curvature. You can't go too far. And also, areas, volumes grow a bit, and then you can grow over the volume of the sphere, of the ball. Okay, and this is how it looks in an algorithm. You see the geodesics of the units. Okay, so the opposite case is that of hyperbolic geometry. This is a drawing by Escher. It's a model of the hyperbolic plane. And here, this is a fishbowl geometry, as Poincaré called it, because if you look at the fishbowl, the classical cruel things with a small fish in a bowl, if it's in the center, you see it at the right size. The closer it approaches the glass, it gets bigger and bigger. It looks like a Jaws kind of horrible thing. And that's what happens. All these fishes are the same size. They don't look the same size, but they should be supposed to the same size. The circle is at infinity, so we have many infinities, not just one infinity. And the volumes grow exponentially fast, so grow distances. And this is also good for us because we like to do something, play all day long with something which grows exponentially fast. And we like to call it the internet. Okay? That's this kind of exponential growth phenomenon. Okay, so what kind of Ricci curvature? So there are two kinds of Ricci curvature which are famous for networks. One is called Olivier Ricci curvature, and it's famous for two reasons. One, it's cool. It has a nice picture actually. B, it has beautiful mathematics, but that won't probably convince you. The advisor of his guys, one of the advisors was Gromov, if says something, anyhow. So it comes from it has a good pedigree. But it seems very cool, and it starts from an observation that in the plane, the balls or the points of the circles are as far away in the mean as the centers. But on the sphere, they are closer, and on the hyperbolic plane, they're farther away. If you want me, I'll go back to the sphere. Sorry. So if you look here, small circles here and here, they're much closer, the points on the circles on the colors, on the polar circles. Then the centers which are the north, respectively south. So that's his idea. So formally we define it in this manner. So it's a beautiful thing. He invented it for Markov chains, which are of course important in biology. And by the way, and the idea was that you want to compare the distance between the centers, the cost of transporting a mass from a point to another. W1 is the cost of distance. How much does it cost? I want to build a pyramid, and I take stones from here and put them there. How much does it cost me? How many hours? How many people I have to kill? How many gallons of gas do I have? Whatever. Yeah, so that's is a transportation cost. And this is a very nice picture. People like it. People try to apply it a lot, but it looks cool, but it's hard to compute. It's hard to generalize to higher dimensions, to more complex structures. It doesn't see hexagons or higher order faces, but it's still good for something. So for instance, it detects the backbone of a network. This is an email network, a communication network, and this is one of the first things you did. And you see the red one is a backbone, it looks like a tree. Like think of the Christmas tree. This is the backbone of the core of the network. And you have these clusters, the local computing centers with which are these red, bluish blobs. Like in a Christmas tree, you have these decorations, yes? Okay. This is the same picture, but cooler, I think. And you see better the blobs, you see, clusters of positive curvature. Okay. And there is another one, which I prefer, which was devised by a guy named Bioinformatics. And it encodes how fast geodesics disperse. It's easier to apply to a very large class of objects which are called weighted cell complexes. Okay. And these include graphs, even weighted graphs like this one. This is a graph, of course, computers communicating, and you have weights, throughput, and power, computational power. You have weights on nodes and the edges as well. Okay? So this is one kind where you can apply it one setting. The other one is, this is David, but it's a graphic David, and it's made up from triangles. So this is also a polyhedral complex made up of triangles. And there is another kind of polyhedral complex, which I choose this picture because it shows how far I can go far away from these things. You see, it's just a pixelized image. Imagine of all the internet. You guys imagine of the internet. Not everybody can imagine when picture would appear line after line after line, or this way. So, and you still can compute its curvature. This is a ladder, by the way.
Yeah, that's the famous girl with a hat.
Yes, 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 it is.
Image processing.
Yes, yes.
Blur your eyes, you can see it.
Yes.
That's a famous picture.
Yes, it's a famous picture, it's a famous story.
Yeah.
Right.
Just go blur your eyes and you can see it.
And since my wife is also Lena, so it's easier for me to use. I'm more inclined. Yes. From this, if I look this way, in Lena, I see her. Yeah. Okay. But anyhow, thanks for. It's famous. She was invited at one of the big congresses.
It was in every textbook.
Yeah, but now she decided she doesn't want to be on this.
No, there were problems with it.
Yeah.
Okay.
When you talk in particular, I tell you where they got the picture from. But the thing is, she was like honor invitee at one of these guests of the biggest imaging conferences in the world, the biggest one. Was every few years, and did one, and she was honored guest, and because they said we made so much, we published so much because of you, you should be here.
Yeah.
Okay. So okay. So you have a formula. So this formula, I mean, you might be frightened by it, but I say no. It's a simple formula, guys. It's just adding, subtracting, and some square. That's elementary mathematics. You don't have to understand how I obtain the formula to write a code for it. And what comes in are just the weights on the edges and then the nodes. So I think it's easier to understand it if you look at this picture. What I need, I have a network like this one with the computers. So I have weights on the nodes, I have weights on the edges, and I plumb them in and I compute something. Okay, so this is like this. Okay. It can be a little bit more complicated, but locally it looks like this. So again, this captures the, not like Olivier, this one captures the dispersion of geodesics. Why? Because if you take combinatorial weights, I mean just counting weights, one for each edge, one for each node, you get this thing. So basically you see the sum of the degrees, how many edges come in, how many edges go out. So deg V1, deg V2 means how many edges come in. I don't know if this works or not. So how many edges come in here and how many edges go out here. So it's a flow of whatever, information, chemical reactions, I don't know. So of course, people, when I started talking about this, we knew we want to look at edges, but people were used to nodes. So you can go from edges to nodes. Just naturally, just take the average. If you have 7 edges, each with this curvature at the node, just do the average, or just the sum. And this is what is called, so you can go from edges to nodes. This should be called proper scalar curvature. That's just for connoisseurs, not for, it's not necessary for us to. Well, and this is a slide which I debated with my wife if I should include it or not. But just to get you feeling better that I'm not cheating you. So the feeling is, okay, why should I complicate my life so much with something like just degree or clustering coefficient? And it turns out that this is a good example, simple example of 2 graphs which have the same degrees, but very different curvatures. The degrees are 2, 2, 1, 2, 2, 2, 1, 1, 1, but curvatures are different. Okay, so this is really something different. And let's go to some examples. Hopefully, not much mathematics for now. So this is a first example we published. I guess it's a PDZ domain mediated protein-protein interaction network, and you'll explain me what it means. But the nodes are bigger and the edges are thicker, the higher the curvature is in absolute value. Red means negative, green or blue means positive, so you see it's generally negative, this one. So it's interesting to picture, but doesn't say too much. But if you look at the histogram of the curvatures, then you can start learning something. So statistics is good for something. Please look here. Have 2 pairs of networks, 2 natural networks and 2 man-made. The natural one are a gene expression network and a brain network. FMRI, okay? And the other 2 are, one is a Google Web Graph, which is very cool, 1,000 pages. And the other one is a word occurs in books. People study that. I remember we were studying, taking Balzac and trying to discover something, how, of the Bible, they do that with the Bible as well. So you will see there is a huge difference. The histograms of curvature is a man-made, are man-made. So they have this structure, it looks like model, so-called model networks, they have this, you see, they have no humps. They are all the exponential of the Gaussian distribution kind of network. The other ones have humps. So this, we discovered the natural networks have humps. Why? So let's see another example. So this is an example, remember the brain network, this is it. This was fMRI. So the people were, the task was very simple. You lay in the fMRI machine for 10 euros. There was a guy who stood there for 200 times just to get the money. I wouldn't recommend that. You attract metal on street. Yeah? But so the task was to clap. So of course the visual cortex is excited. So why do we have 2 humps? Because people have the bad habit of looking at their hands when they. So it's not that the motor one, but also the visual one. And you have some maintaining, the peak there is. You still breathe.
So if you close your eyes, yes. You get one.
Yeah. Oh, I guess you still have something associated with people picture it in their minds. I don't know. That's a good question. It's an excellent question. Thank you so much. It's a very nice one. So we applied this to try to understand better autism, to find more centers associated with. And this is there are good results for both Olivier and Foreman. I won't get into that. That's a talk by itself. And you can use for metrical imaging. For instance, you wanted to do this automatic segmentation of the ossicles, which are the smallest bones in the human body. It's very hard to see them, to serve them. So we did this with Richie, and these are 2 classical methods to see far less resolution. And I have to say that before doing networks, I did imaging, and Ricci works like a charm. But okay, so then I bring this result without much explanation. We did the same thing for stem cell differentiation for correlation networks. And as you can see, we have two plots of curvature and of entropy, and they correlate extremely well in stem cell differentiation. Ricci curvature, graph Ricci curvature, the one you saw goes down. As you go down on differentiate more, I don't know how to put it, and so does entropy. And they're very well correlated, and that was the first time that somebody observed this relation. And for cancer, we have the opposite phenomenon. The more cell gets out of normality, closer to being cancerous, the more the entropy grows. And you can see this in these histograms. Okay, so, and there are other type of networks, the hyper networks. So beyond networks, there are hyper networks. There are a few types of hyper networks. One is the simplest one, these chemical reactions. So they have got a paper in Nature for doing this kind of stuff. They are very simple curvature ones. You just reactants A and B, put them in reaction one, and you have got stuff that is C and D, and so on and so forth. So you have these huge trees, basically. They're kind of boring mathematically speaking. So histograms, of course, they have humps, just very peaky humps, because the graphs, the structures are very simple. But more interesting networks are those who have higher order interactions. So we three might know each other. I mean, Lika, Minja, and myself. We might talk to each other, but this doesn't mean that the three of us have a paper together. If three of us work on a paper, then we put a full triangle. If we four work together, there's tetrahedron, and so on and so forth. So in fact, hypernetworks can be modeled by this polyhedra. And this is an example which you might find more familiar. I gave a social example one. This is one from protein interactions. There were two proteins interacting, means an edge, but if three proteins come together to produce something, to control something, then you plumb in a triangle. If you have four, then a tetrahedron, and so on and so forth. So it can get a bit more serious than that, but never mind. So of course, then you need a formula. So the formula is very similar to the original one, but a bit more complicated. Why? Because in a network, in a graph, you have edges and nodes. That's it. Now I have nodes, edges, and faces. So I have to take all of this into account. So all the signs are more or less the usual ones, except there is a sign there which means that things are parallel. So when things are parallel, what does it mean? Well, it means what we expect. That they, for instance, here these two red edges are parallel, whereas all these edges are not parallel. Why? Because parallel means you have a common parent or a common child, but not both together. I mean, either you have two, like here. So you see these two red edges are edges of the same face, of the same parent, but they don't intersect. They don't have a common vertex together, yes? Whereas these two, for instance, these two, this one and this one, belong to the same face and also intersect in the same vertex. So they are not parallel, okay? Okay. So it's, if things look parallel, they are parallel. And of course, in a triangle, you don't have parallel edges. So it's kind of easy here to see it here. Okay, so having said that,
I have a question.
Yeah.
So go back. So that formula you had, this is for three-way. You have triangle.
No, you can
Any general.
Any general. Yes. So for instance, here you see the picture is as a quadruple.
Okay.
It's okay. And what's more important, well, that's something what you might say, okay, you have this, you might have a formula which is very complicated for any dimension. Yes, I have formulas for that, but I'm not interested because the Ricci curvature, if you remember, lives on directions, on vectors. Vectors are edges in graphs, but edges are only the intersection of two faces, of triangles, quadrilaterals, and so forth. I'm not interested in higher dimensions. So all the information lives at the intersection of, say, triangles or squares, nothing more. So it's a cool thing to do. Yeah.
Can you go back one slide? I meant to ask you earlier too, the W's were still Wasserstein distance?
No, it's omega's weight.
Yeah,
Wait.
Weight.
yeah.
So, okay. So one thing we use omega, not confuse it, but let's it hard to confuse it. But Wasserstein distance is capital W, always capital. Notation is problematic, but it's not mine. So I'm half guilty. Okay? Yeah, I understand. So these slides and what the discussion, previous discussion would have made more sense before what we discussed, the example which I gave you with melanoma. But I thought first I give a technical example, then I explain how. So we thought about the Wasserstein landscape. So if you are high in the Wasserstein before differentiation, you have a high entropy, and the more you go down, entropy goes down. But you can see, so the question is, at least I ask myself, but it's not just me, many people before and after, it's okay, it's a nice model, it's a beautiful metaphor, but is it just poetry or is it perhaps something we can compute? Well, I say, and many say that yes, we can do that, because if you see this path going down, it's just basically a graph, an oriented graph, oriented network. So if I have an oriented network, you might think anything else, but when I say an oriented network, I think curvature immediately. Yes, it's conditioning. Yes, it's also biology, Freudian conditioning. So the way we introduced this approach to biology or to quantization of cellular trajectories and pluripotency were Wang and allies. I don't remember the names of the papers. There are two different papers. Yes, and the idea is that you look at entropy. And you see that this is a bad image. Next time I should improve it. So if you are upper in the Waddington landscape, it's more promiscuous, less certainty, higher entropy. The lower you go, you are more committed, higher certainty, lower entropy. And so, as we said before, it decreases entropy with cell differentiation and increases is cancer, because cancer is taking things out of order, if you wish, in a sense. Okay, so is there a relation between entropy and curvature, and Ricci curvature more precisely? Yes, there is. They are both local. They are correlated by this relation, and this is related to robustness. How hard is to break apart the network? And this was something that Alan Tenenbaum and his collaborators discovered first. And so why use curvature and not entropy? Well, one of the reasons is because I understand very well the geometry of curvature and can compute it easily, whereas entropy, the physics kind of complicated. And also, it's harder perhaps to compute. So what kind of entropy? Ricci curvature. So at the beginning, they used Olivier. Why? Because that's what they knew. But we saw, well, we didn't see, but I stated it. And I'll ask you to believe me that it's very, very, very hard to compute. I mean, you have to do a lot of optimization and still crashes computers and these laptops. So the trick is that Ricci curvature is a Ricci curvature, so I can do it with Foreman instead of Olivier, because it's easier to compute. Basically, they give the same results. They're very strongly correlated. As opposed to Olivier curvature, I can easily extend this to higher dimensions, and I'll talk a bit about it.
So that's typo. It's extendable, right? You're not,
Ah,
yeah. So
yeah, it's
that's
a,
okay.
yes, it's a type
It's
of,
extendable.
it's
just
kind of
typo.
funny, yes,
Word
it's-
thing, yeah.
it's not expandable. That's a movie, but. it's kind
Uh-huh.
of- Sorry, yeah,
It's a
thank you,
spell
for-
It's a spell checker.
I think he tried to correct me and
Yeah.
he corrected me in the wrong direction. Yes, thank you so much. So if you use this, then you are very much into considering hypernetworks for this kind of phenomena. Okay, so I'm going back to what we saw before. The same examples basically, just now we're taking into account not just edges, but two faces, triangles, and so on. In fact, just triangles, because looking at higher order faces, it's kind of complicated. So as you can see, again, you have the two-dimensional Ricci curvature, the new one. You have for different types of cells. Yes, two-dimensional, one-dimensional, two-dimensional, and here you have entropy, and we see the correlations which are the same. And the same phenomenon happens when you go in stem differentiation, how these things evolve when you go down in the Waddington landscape. There is an explanation. If we want, you can read the paper. The same thing we did it. So there was, so the first paper on edges was, it's melanoma, if I remember correctly. The second one, Kevin, my co-author, wanted to this with sarcomas, and you see basically the same phenomena happens. And it takes some time to look at these figures and to understand. And it's a bit of noise, of course, as you know, in biology. But the best proof, in my opinion, is this result, which you look at genes which are relevant. So we have how many genes, significant genes we can discover. So the blue circle is the formal Ricci one-dimensional for graphs. The second one, the acrylic purple one, red one, whatever, is the genes we discovered with two-dimensional, the higher order Ricci, formal Ricci curvature. And the lower yellow circle is a classical differentiation gene expression. And as you can see, any of the one-dimensional, any of the formal curvature discovers much more. There are intersections, of course, but the curvatures discover much more relevant genes than differential expressions. And moreover, the two-dimensional one that takes into account higher order structures is even more efficient. Okay, so for me, I have to presume that computations were okay. So this would say that yes, it's worthwhile looking at these curvatures. Okay. So again, the one-dimensional, the graph, the network is just a skeleton of the higher order structure. So it captures the essential phenomena that misses some. That's why you get more in 2 dimensions. Yes. So again, you have the same differentiation. This is conclusion, which I spoke about just before. You get, you can distinguish undifferentiated stem cells from the committed ones, differentiated ones, and cancerous cells from the healthy ones. Okay. So we think it might be useful or something. Okay, so these are the papers we published on. Okay, so now before, I won't take up all your time, I kind of rushed it, but still a few things. So this is something which I learned from Indica. It's very nice and dandy to talk about correlations, but they're somehow artificial, yes? So you would like to look at physical interactions, and this you can look at chromatin structure, chromatin structure. So this is one of the figures which I borrowed from you, as you know. Another one, so how it comes, you have this special, so they, in space they come close together one to another. They come close to each other in space. the genomic loci. So if two come together, then you have an edge. Paint them as an edge, represent them as an edge. If three come together, you can paint them or represent them as a hyperedge, like here, three together. That's one way of looking at it. The other thing is looking at them as a triangle, as we did before. So it depends on what you want. If you want to do set theory, or to be cool and say I do hypergraphs and everybody thinks he knows what it means, we'll do this. But when you try to compute something, you represent this as a triangle. And there is a standard way discovered by a guy named Ethan Block, who shows that you start from these kind of objects, very abstract ones, and you can produce a standard simplicial complex, which has a natural form and rich curvature. So that's the right way to do it. And the other way around is very simple, to start from a polyhedron and produce these kind of structures.
Can I ask a question here? What are the difference between the core interactions and non-core?
And that's a question for Indica.
Yeah, I mean,
You define the core. What's the yeah?
So core, okay. So if we look at, so in a core, as if you remember, that would give you, there are many ways of defining a core. But if you remember the example which you used, I brought in the beginning, the red Christmas tree with the green globes. So the red one is a core. It represents the main part of the network where most of the communication, most of the connections happen. If you talk about information, where is the information flows faster, so farther. And also this way you can, because it's a main thing, it's easier to disconnect if it's attacked by something. Let's say by a virus in the.
So you're thinking of computer network.
Yes, I'm thinking about a computer network.
Yeah.
So the trick is how to think about it for biological networks. So one way, which is kind of artificial, is to look only count how many edges come at each node. And that's only combinatorics. So good definition of the core is to look at those regions of the network that have the densest node population with the most edges coming in and the longest communication bridges between communities. So the densest, biggest communities, densest packed communities is the longest connection between highways. Okay, so if you think New York, Boston, Washington, DC, Baltimore, they are part of this. Okay, whereas, I don't know, Yonkers is less. Okay, so big network, big clusters with strong connections. Okay, so so how you discover them? It's easy to count how many things come in, but how you discover the long connection? How do you do this correlation? How you discover both of these phenomena together? The answer is straightforward, doing Ricci curvature. We saw it works. So we do, so now the question is how to define Ricci curvature for higher order structures, and that's what I'm proposing here. This work in progress. And on the biological side, on the mathematical side, we have some results. Okay.
Yeah,
Okay.
it's helpful. So now I gotta translate this into Chrome.
Okay, so now you have this, somebody did that for me. Again, Indica's team. So now you look at, you have some core chromatin in chromosome 2, and you look at edges, and you look at triangles, and the quadruples, and so on and so forth. But, so each picture means it's another type of phase. But of course, in a real life network, in a real chromatin, they won't come like this in different colors or different layers. They come together. So this is I took a picture of indicas and I jiggled it a bit to show you how you build them. So you will have some triangles, and you have some quadruples, some pentagons, but they don't live separate. They're a whole family. They live together. So they come together like this, either nicely only at vertices, or along edges, or along diagonals. So your hypernetwork, chromatin hypernetwork, will be this mess made up of many, many phases. Okay, so this is a small scale, but if you want to look at a real life example, again, Indica saves me. Here it is. This is a small portion of chromatin hypernetwork in chromosome 19, it turns out. And what I didn't notice before is that you have these faces which are also the transcription factories, which are kind of painted differently. And you can see that a real life network, hypernetwork, a real life meaning chromatin hypernetwork, is not just composed of very different types of faces, not just triangles, but in fact, there are higher order ones. We saw in the work with Kevin and Romy Sandu, which you saw before with the and stuff. Then you have faces of very high orders. Nobody really knows. It's very hard to compute something, and nobody knows how relevant they are biologically speaking. Yes. So usually people restrict these main. Most things happen in 4 perhaps 4 or 5 phases. So it's it's not uniform at all. It's not just the same type of phase. It has many holes. So some part of it is almost tree-like, almost graph-like, and some of it is quite dense. And so usually people who do such stuff say, oh, I need something uniform, I have to presume this and this and this and that, and I would like to do nice standard geometry. I don't need that for formal curvature. And anything goes in, as long as I have it, I can produce result. It really computes formal curvature for each edge. It doesn't matter for the apparatus how dense this or how uniform the network is. Okay, so now we did that for another piece. We have, unfortunately, we are a bit late with this, but in any case, you can see here the computation of the main curvature of nodes. You can see intensity by how heavy a node is, how high or low is the curvature, and for edges as well. The blue line, there's one linear sequence on the chromosome, which means, of course, something for you guys. Oh, sorry. Okay, so where am I going? Okay, so again, we did histograms. So here is the histogram. And as you can see, we have this is a node-wise and the hyperedge curvature. And you can see there is a hump effect. And Sarah, who did this, but it's very spiky. Yes, this is a good sign. It means, because these are just combinatorial ways, just counting. So it means that basically, okay, my network might be kind of sparse and uniform, but still most edges have essentially the same number of faces around. Most of them. Yes? That spike means that most have the same number of edges around them, which says something about this chromatin structure already, even without weights, even without anything more. So I'm going to finish in a few slides. Okay, I still have a few minutes.
3 minutes.
3 minutes.
Yeah.
That increases my entropy very. So we think so you have immune networks, which are also networks. So we think how to introduce good hyper network structure, and how to explore curvature for their understanding. It's another project, and you can go back to the Waddington landscape. I've now brought a picture which I brought different pictures for different occasions. I like to change clothes, so to say. So we would like to understand through curvature the geometry evolution along the Waddington landscape. So there is a new trick. You use it in Waddington landscape, and also in cicatrization. You just don't use just mass transportation. So think of it on cicatrization. You have cells, and you bring cells from everywhere, and you bring them to the wound, but they have to increase in number, otherwise you don't fill in the wound. Yes. So you just don't just transport mass, you modify the mass. So this is a general and optimal transport, and that's what I concentrated lately on. And I have a new kind of Ricci curvature for hypernetworks, but I won't bother with that much mathematics. Okay, so thanks for your attention. questions. Sorry, and sorry for the rather impressive presentation. It was kind of fruitful, I know, but try questions. It should be question, another question.
Yeah.
One question would be if I'm the John in cash. Yeah. Please.
I can start.
Let me take my.
Yeah, very nice talk, interesting. So I wonder, all of your curvatures and methods, how does it scale with the number of hyperedges and the size of the hyperedges?
I have the number somewhere. So for hyperedges, it's, I couldn't say, because there are two versions of that I'm aware of. One proposed by Asude Evans and another guy who take the mathematics and transportation and try to translate it and things instead of taking things in our path to take things from faces and volumes to faces and volumes of whole mess. Mathematics is beautiful, I don't know of applications. Melanie Weber and myself have a version which is very simple. You reduce, going to the dual graph, reduce it to a one-dimensional case. If you want to complexity, I can send you that. I have it somewhere.
Yeah.
It's
so
horrible.
We're working on some similar problems too, with the hypergraphs and chromatin structures. But we sometimes see in sprite data that the hyperedge of size 1,000 or more, and that doesn't really scale with our methods.
No,
the
it doesn't.
optimization.
It doesn't. Nobody. I mean, so we had 16,000 or something like that, and in the end we just look at triangles and quadrilaterals. But I think Cooper discovered that essentially all the main things happen for low order fields.
Sometimes, but I
Okay,
think
yeah,
the
yeah.
sprite data says that interchromosomal hubs are very important too.
I'm sorry, it's very bad.
Yeah, I understand.
Forman is much more friendly from that standpoint.
Thanks.
I wish I could be more.
Any more questions? Emil, I still have that question that even the Olivier, this curvature compared to like high order hypergraph centrality measures, like eigenvector centrality.
So,
These are very fast computable.
yes, so Olivier is bad for that.
Yeah, but
Yeah,
these are fast computable,
Yeah.
the eigenvector centrality for
Yes,
hypergraphs.
they improved it a lot.
Yeah.
Because it's challenging. And they like it because it's very expressive. There is a lot of deep mathematical theory behind it. So many people like it. You saw that things with the bridges, with the internet. I'm one of the authors. So yeah, in the beginning, I was very much do it. And then you see that it. Thank you. Yeah.
You're standing for a
Well, I'm just kind of curious about, you talk about, how many petaflops are we talking about here? I mean, how does this scale a computation?
Oh,
I mean,
again, I don't remember.
Yeah.
I asked a friend of mine who specializes in that, and she said me, if you want it, I'll send it to you. This is kind of, format is kind of simple to understand how it works. Once you do the counting, then it's squared or something. So basic computation is squared, of course,
It's kind of
find
mathematical transform that allows you to, like the Fourier transform, it's
Yes.
a transform.
okay,
Is what I'm seeing you
yeah,
say.
you're right.
The transform gives you the ability to do a computation that's tractable and then back transform it into something that you can.
Sure. Beautifully
I mean,
put.
that's
Thank you
really
very
what
much.
I'm getting at.
Yeah, it has another advantage which I didn't use here. You have this flow associated with it, so you can predict how things evolve in time using curvature. But that's another. It's not for just one.
Okay, very good. Any more questions, students? No. Okay, thank you so much, Emil. Thank you so much. Yeah.